Hello, today I'm going over cell membrane lecture. So we're just introducing the cell parts, cell organelles, and getting to know the general structure. So this is just the cell membrane or the skin of the cell. The cell membrane acts as a protective boundary for a cell composed of a double layer of specialized molecules called phospholipids. We see these here's one phospholipid and here's two layers of phospholipids. Each phospholipid consists of a polar charged head made of a phosphate group in glycerol and two nonpolar tails made of fatty acid chains. Because the heads are polar, they are attracted to water and can form hydrogen bonds, while the nonpolar tails repel water and are attracted to each other. So you'll see the head is always going to be on the outside of the cell and the inside of the cell because there's water, H2O, in both of these areas. Inside of the cell where the tails are, there is no water. To accommodate these properties, phospholipids arrange themselves into a bilayer that resembles a sandwich. The polar heads act as the bread, forming the outer and inner surfaces of the membrane to interact with the watery environments inside and outside the cell. So, similar image down here. These are the heads on the outside and on the inside. The nonpolar tails serve as the filling, tucked safely away in the center of the membrane where they are shielded from water. So again, H2O outside the cell and inside the cell, but in the skin, in the middle of the skin area, there's no water. These are all called hydrophobic. They do not want to be near water, the tails. This specific arrangement allows the membrane to effectively control the passage of materials into and out of the cell. The cell membrane is defined by the fluid mosaic model, which describes a flexible and dynamic structure rather than a rigid one. So, fluid mosaic model, that just means that this skin can move, it can stretch a little bit, it can bend a little bit, it can change its shape rather than be solid and not moving. This arrangement allows individual phospholipid molecules to move laterally and slide past one another, behaving much like a fluid film of oil. So that's the red stuff in here, right? These are the phospholipids. They can move around each other. While the phospholipids maintain this fluidity, the embedded proteins are anchored in a specific orientation and do not flip vertically. So we can see these proteins in blue here, and they're going to stay in the same orientation top to bottom. They might move side to side a little bit because you have changing phospholipids, but generally they're stuck in place and very much stuck in this orientation, top and bottom, okay? The mosaic portion of the name refers to the diverse variety of molecules that stud the membrane, creating a complex pattern of different textures and functions. So everything we see here has some kind of purpose and function. We haven't gotten to it all, but everything that's part of it, that's the mosaic part. It just means there's lots of different pieces doing different things. Various specialized molecules are embedded within these phospholipid layers to provide the membrane with unique biological properties. Cholesterol, which you can see here as in the yellow, cholesterol molecules play a vital role in structural integrity by strengthening the membrane, while different types of proteins facilitate the transport of materials or support the cytoskeleton. Furthermore, carbohydrates attached to these membrane proteins act as critical identification tags that allow cells to recognize and distinguish one another. So you've got let's see, carbohydrates here, these little branches that can communicate with other cells or send messages to and from other cells. You've got your embedded proteins, which we already talked about. That's in the blue. These diverse components work together to ensure the cell membrane remains functional, secure, and communicative within its environment. And then we just have another picture of that same portion of the cell membrane. This has identified a few more of the things that we've discussed, like the cholesterol here and the carbohydrates and the proteins. Okay. The cell membrane possesses the vital property of selective permeability, which allows it to control the movement of materials into and out of the cell. So selective permeability, that means it controls what goes in and out of the cell. Some stuff is going to move in and out in different ways, but this barrier can dictate what goes through it. This characteristic ensures that only specific substances can cross the barrier. By regulating this traffic, the cell effectively maintains homeostasis despite shifting environmental conditions in the surrounding area. This precise control protects internal chemical reactions from being disrupted by sudden changes in external ion concentrations. So stuff has to pass in and out of the cells. Sometimes it's products that are made in the cell that have to pass out of the cell. Sometimes it's a chemical message from another cell that has to get into the cell to pass its message along. There's a variety of reasons that things would be going into and out of one particular cell. The method a molecule uses to cross the membrane is determined by its size, polarity, and the concentration gradient between the interior and exterior. Small non-polar molecules can easily slide through the phospholipid bilayer. So they're talking like this here, just going straight through. Whereas small polar molecules typically require the assistance of specialized transport proteins. So this right here. Large substances that cannot fit through these channels are instead moved across the boundary using membrane-bound sacs called vesicles. That's this. So there's a vesicle inside of the cell that has some chemicals here, and it's going to merge with the cell membrane, because the vesicle is made up of the exact same stuff. And when it merges, it can open up and release those chemicals outside of the cell. Large substances that cannot fit through these channels are instead vesicles. Depending on the specific molecule and the direction of travel, the cell may either allow passive movement or expend energy to force the transit. So passive, it happens on its own. Expending energy means energy input in order to make it happen. All right. I hope this helps get us going. If you have any questions, let me know. Thank you very much.